Dr. D, 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 Dr. D. Explain stuff.
Hey everyone, Dr. D here. In this video, we are going to be covering Chapter 5 of our Bauman textbook, Microbial Metabolism. So let's go ahead and get started. Remember, before you actually watch this video, if you don't recall how cellular respiration works, or if you don't recall how photosynthesis works, please watch the videos I have attached here. There's a card that should be up here at the top right of your screen. There should be a card right now for cellular respiration, and then right after that I'm going to flash a card for photosynthesis. Please watch those videos first, because this is a second year course and cellular respiration was explained in Biology 1406. So if you don't recall how cellular respiration works, make sure you watch my video on cellular respiration. And like I said, make sure you watch the video on photosynthesis as well before you attempt to watch this video, because I'm going to assume that you remember how cellular respiration works for this course, because it was taught in detail last semester. So if you've already done that and you're back and you've already refreshed yourself on what cellular respiration is and what photosynthesis is, welcome back. Let's go ahead and get started. So in here, we're just going to review it very briefly, and we are going to talk about some of the differences between cellular respiration in eukaryotes versus prokaryotes. So let's go ahead and get started. So recall that carbohydrates, fats, and proteins are all consumed as fuel. And of course, you get the most amount of fuel from fats, followed by carbohydrates, and followed distantly by proteins. Now, cellular respiration is the process of breaking down those energy molecules. Specifically, with this equation here, you're breaking down glucose and using the energy in the sugar to produce ATP, right? To produce ATP energy. So recall the equation: 1 glucose, C6H12O6, plus 6 oxygen are used as reactants in this process, and the products are 6 CO2 molecules, 6 water molecules, and energy in the form of ATP. What you need to know is that there are 32 ATP net NET, right? Net 32 net ATP for prokaryotes, and only 30 net ATP for eukaryotes. All right, just recall that. And of course, heat is given off as well. Now, you should remember from before the terms oxidation and reduction. Oxidation means loss of electrons. Reduction means gain of electrons. And the whole process of cellular respiration involves a series of redox reactions, reduction oxidation reactions, where electrons go from this molecule to that, to that, to that, to that. Electrons are moving from donor to recipient, to recipient, to recipient, and that's how cellular respiration works. And also that's what fuels photosynthesis as well, these redox reactions. And remember, the electron carriers have names as well. You've got NAD plus and FAD. Those are the electron carriers involved with cellular respiration. And then you have NADP plus, and that's the electron carrier involved in photosynthesis. So again, this is a overview of redox reactions. Remember, you have an electron donor. For example, this could be glucose with all of its various and numerous electrons. the electron donor donates an electron to an acceptor. For example, what if this was NAD+, right? What if this was an electron carrier? Well, then the sugar would lose an electron, which would mean the sugar is oxidized, and then the carrier would gain an electron, which means the carrier would become reduced. Okay, so the carrier is reduced and the sugar is oxidized. Okay, so remember that when we're dealing with cellular respiration, you're going to be converting ADP to ATP. So you're phosphorylating ADP to ATP, and you're using the power of sugars, fats, and proteins in order to do that. Okay, so how do we do that? There's cellular respiration and there's fermentation, right? What was the difference there? Do you recall? So let me show you. During cellular respiration, remember, respiration means that you're making ATP from sugars, and you're doing it with the help of glycolysis and oxidation of pyruvate and Krebs cycle and the electron transport chain. Did you guys follow that? So when you hear the term respiration, that means that you are involving glycolysis and oxidation of pyruvate, which is this step between glycolysis and Krebs cycle and electron transport chain. Respiration involves all of these processes. Okay, and you get a lot of ATP this way. Fermentation, on the other hand, does not involve the latter phases. It only involves glycolysis. Did you guys follow that? So fermentation only involves glycolysis. So fermentation only yields to ATP, whereas respiration involves glycolysis, oxidation of pyruvate, Krebs cycle, and the electron transport chain. So you're going to get a lot more than 2 ATP. In fact, if it's oxidative cellular respiration, which means cellular respiration where the terminal electron acceptor is oxygen, well, in that case, you're going to end up with 32 ATP for prokaryotes and 30 ATP for eukaryotes. But remember, you can also have what's known as anaerobic cellular respiration. What does that mean? That means that the final electron acceptor, the terminal electron acceptor, is not oxygen. Instead, it is some other highly oxidative molecule, either a sulfur-containing molecule, a nitrogen-containing molecule, or some other molecule that's highly oxidative. And these creatures do not use oxygen as their terminal electron acceptor. They use a different molecule as their terminal electron acceptor. And those creatures usually make less than 30 ATP. So here's an overview of cellular respiration. First, remember respiration? Respiration means you've involved glycolysis, you've involved oxidation of pyruvate or pyruvate oxidation, however you want to say it. You have the citric acid cycle, also known as the Krebs cycle, which is also known as the TSA cycle. Okay, so you have those. And then you also have the electron transport chain and what's known as chemiosmosis, which involves ATP synthase. Okay, together, electron transport chain and chemiosmosis together is known as oxidative phosphorylation. So again, cellular respiration involves step 1, glycolysis, step what I call 1.5, pyruvate oxidation or oxidation of pyruvate, step 2, citric acid cycle, or also known as Krebs cycle, or also known as TSA cycle. That's step 2. And step 3, known as oxidative phosphorylation, which involves 2 main parts, electron transport chain and chemiosmosis, which occurs through ATP synthase. Okay, this is known as aerobic if the terminal electron acceptor here is oxygen. And this would be known as anaerobic cellular respiration if the terminal electron acceptor is not oxygen. Does that make sense? So if the terminal electron acceptor is not oxygen, but there is a terminal electron acceptor here at oxidative phosphorylation, then it is known as anaerobic cellular respiration. So again, I want you guys to know this. This is very, very important for you to understand. Respiration means, cellular respiration means, you have step 1, step 1.5, step 2, step 3. What does that mean? You have glycolysis, pyruvate oxidation, citric acid cycle, and oxidative phosphorylation. So what's aerobic cellular respiration means? It means you have glycolysis, pyruvate oxidation, citric acid cycle, oxidative phosphorylation, and your terminal electron acceptor is what? Oxygen. That's what aerobic means. So what is anaerobic cellular respiration? What's anaerobic cellular respiration? That's right. You've got glycolysis, you've got pyruvate oxidation, you've got citric acid cycle, and you've got oxidative phosphorylation. However, your terminal electron acceptor is not oxygen. It is some other highly electronegative molecule, if that makes any sense. Okay, so this is an overview of respiration. This is actually a really nice overview of aerobic cellular respiration, which makes how many ATP? Do you guys remember? That's right, 28 plus 2 plus 2, 32 ATP. In what? In prokaryotes. 30 net ATP in eukaryotes. If you're wondering why, because the 2 ATP that made here during substrate level phosphorylation, well, 2 ATP are used to get pyruvate into the mitochondria in eukaryotes, so you kind of lose those 2 ATP. You have to spend them to get the pyruvate into the mitochondria in eukaryotes. Prokaryotes, on the other hand, they don't have a mitochondria, so you don't have to spend this 2 ATP to get the pyruvate into the mitochondria. Hence, you net 32 ATP. All right, so let's do a quick overview of glycolysis. Again, if you don't recall glycolysis, you don't recall cellular respiration, watch those videos I referred to earlier, the links above, because that will help you out a lot. Okay, but so I'm going to quickly go through these 10 steps of glycolysis. Okay, I'm going to go through these right here. Glycolysis. You have a 6-carbon sugar called glucose, right? Each of these pink dots represents a carbon within the sugar. You don't need, by the way, you don't need to know every one of these little steps I'm going to address right now, but I'm going to explain them just so you have an understanding, but you don't need to memorize every single step. So glucose gets phosphorylated into glucose 6-phosphate. What does that mean? An ATP comes along, and the terminal phosphate, the 3rd phosphate of ATP, is given to glucose. So glucose picks up a phosphate group from the ATP, and now ATP is converted to ADP. Why? Because it had 3 phosphate groups. It gave 1 phosphate to glucose. Glucose now picked up the phosphate group, so now ATP has become ADP. It only has 2 phosphate groups. Does that make sense? And that's what this term phosphorylation means. When ATP gives a phosphate group to glucose, that means phosphorylation. That's where the term phosphorylation comes from. If a molecule picks up a phosphate group, it's been phosphorylated. Does that make sense? So glucose gets phosphorylated into glucose 6-phosphate with the help of ATP. And then there's an isomerization that happens where glucose gets changed around into fructose 6-phosphate, which is an isomer of glucose 6-phosphate. Next, it gets phosphorylated again with the help of another ATP. So now you have basically fructose with 2 phosphate groups. So it's called fructose 1,6-bisphosphate. Why? Because the 1st carbon has a phosphate group, the 6th carbon has a phosphate group, so it has biphosphate groups. Okay, fructose 1,6-bisphosphate. Next, the molecule gets cleaved. But before I tell you about that, I just wanted you to know that because we just used up 2 ATP, This is called the investment phase of glycolysis. We just used 2 ATP. And we're trying to make ATP, so this is quite an investment, right? We just invested 2 ATP to start the process, right? So this is called the investment phase. Well, now we're going to transition into the payoff phase, where we're going to actually start making some ATP back. It's almost like you have to spend money to make money. Well, you got to spend some ATP to make some ATP. All right, so what happens at this point? Fructose 1,6-bisphosphate gets cleaved. That means chopped or broken in half. So now you've got 2 3-carbon sugars. You see that? 2 3-carbon sugars known as glyceraldehyde 3-phosphate. So this 3-carbon molecule is called glyceraldehyde, and on the 3rd carbon you have a phosphate group, and you got 2 of them, right? So that's why you have 2 columns here. You have a column here and a column here. Why? Because you have 2 of these glyceraldehyde 3-phosphate. So anything that happens down this column also happens down this column. Okay, so you've got glyceraldehyde 3-phosphate. It is then going to be oxidized. Think about it. There are hydrogens that you can't see coming off of these carbons, right? There are hydrogens attached to this glyceraldehyde 3-phosphate. You're going to give some of those hydrogens, or actually 2 of the hydrogens, you're going to give to NAD+, the electron carrier. NAD+ is the oxidized form of the electron carrier. NAD+ picks up 2 electrons and 1 proton to become reduced to NADH. And that converts glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. Okay. Why did it pick up another phosphate group? Well, because it becomes phosphorylated again. Okay. Again, if you guys are like, huh? I don't understand all this, what's going on step by step. Again, I'm just explaining it step by step so you have an overview. You don't need to know each and every step of this process. So if you're getting confused, like, I'm never going to remember that glucose 6-phosphate becomes fructose 6-phosphate, becomes fructose 1,6-bisphosphate. You don't need to remember all that. I'm just explaining it so you have some understanding. And then later on, I'm going to tell you what you really need to know. Okay, so don't panic. Just bear with me. So now you've got a doubly phosphorylated 1,3-bisphosphoglycerate. All right. This molecule is then used to make ATP through a process known as substrate level phosphorylation. You're taking this phosphate group from an organic molecule. You see, this is an organic. Do you guys remember what organic molecule means? Organic molecule means a molecule with carbons in it. You see these 3 carbons? Those 3 carbons make 1,3-bisphosphoglycerate, an organic molecule. You're going to hand this phosphate group to ADP. ADP becomes phosphorylated to ATP. Did you guys follow that? So that's what substrate level phosphorylation means. An organic molecule gives a phosphate group to another molecule, okay, with the help of an enzyme. You don't need to know that. Okay, so now you've got 3-phosphoglycerate because you lost one of the phosphate groups. You then do a isomerization to 2-phosphoglycerate. So the phosphate group moves to the center carbon, and then there's some kind of dehydration that happens. And lo and behold, you still have another phosphate group. You then use substrate level phosphorylation again to give that phosphate group to ADP to form ATP, phosphorylate ADP to ATP. So you've made 2 ATP, and that happened to each of these 2 glyceraldehyde 3-phosphate. So you've actually made 4 ATP, right? Because you had 2 of these glyceraldehyde 3-phosphates. So you invested 2 ATP. You made back 4 ATP. So how many ATP did you net? You netted 2 ATP. And how many NADH did you make? You made an NADH from this glyceraldehyde 3-phosphate, and you made an NADH with this glyceraldehyde 3-phosphate. So you also yielded 2 NADH. Okay. Now, what happened to your sugar after the second phosphorylation? What you're left with is a 3-carbon sugar called pyruvate, or sometimes known as pyruvic acid. All right, you guys. So how many pyruvate do you have? 1, 2. All right. So you're like, wow, my brain just got exploded, Dr. D. How am I supposed to remember all this? And again, remember, you don't need to remember everything I just said. What I need you to know about glycolysis, which again you should have learned in 1406, hopefully. What you need to know about glycolysis, you guys, is that you start with a glucose. You start with sugar, glucose. All right, and that sugar is a 6-carbon sugar, which is then cut in half, right? And you're going to end up with what? Is it called glucose when you're done with this process? No, it's not called glucose when you're done with this process, right? So when you're done with this process, it's known as pyruvate. Okay, pyruvate. Pyruvate is a 3-carbon molecule. All right. So what you need to know, you start with glucose, you end with 2 pyruvates. But is that all? No, you invested 2 ATP and you got back 4. So you net 2 ATP. You should know that glycolysis nets 2 ATP. And then is that all? No, we also yielded 2 NADH. So what you need to know about glycolysis is, again, you start with 1 glucose, you end with 2 pyruvates. You also netted 2 ATP. You also yielded 2 NADH. And by the way, you should also know that these ATP were formed by what process? That's right, substrate level phosphorylation. Okay, so hopefully that makes sense. Let me know in the comments below if you have any questions about that. And hopefully this is a review of glycolysis from your previous classes. Okay, so now you know the first part, glycolysis. But what about the next part? Oxidation of pyruvate, right? Or pyruvic acid. Do you remember pyruvate can also be known as pyruvic acid? Well, the next step is called oxidation of pyruvic acid or oxidation of pyruvate. And this is when this 3-carbon molecule, pyruvate, and how many pyruvates did you have at the end of glycolysis? 1, 2. So remember, you have 2 of these pyruvic acids, 2 of these pyruvates. What happens in the next step? Pyruvate becomes cleaved, which means this carbon, the 3rd carbon, leaves as CO2. So you've just released CO2. And now how many carbon molecule do you have left? Well, one of the carbons left is CO2. So you have a 2-carbon molecule, no longer known as pyruvic acid or pyruvate. It is now called acetate or acetic acid. Now, acetate is modified. It picks up a coenzyme A. This is, you know what a coenzyme is. It's an organic cofactor. Coenzyme A attaches to acetate, and acetate becomes acetyl-CoA. Acetyl-CoA. And in the meantime, look what happens to acetate as well. Not only does it pick up this coenzyme, but it is oxidized. So acetate is oxidized, and NAD plus picks up those electrons. So NAD plus is reduced to NADH. Acetate is oxidized to acetyl-CoA or acetyl coenzyme A. So this is what I affectionately call step 1.5. Step 1 was glycolysis, and step 1.5 is called oxidation of pyruvate or oxidation of pyruvic acid. Or you could call it formation of acetyl-CoA, whatever you want to call it. It's all the same thing. Next, we're ready for the Krebs cycle, right? And remember, what were the other names for the Krebs cycle? Krebs cycle, citric acid cycle, TCA cycle, right? Okay, TCA. Did I call it TSA cycle before? It should be TCA. TCA cycle. All right, so again, this is what I just showed you. Pyruvate loses a carbon to be as carbon dioxide. It is now a 2-carbon molecule. It picks up a coenzyme A, and it is oxidized into acetyl-CoA. NAD plus in the meantime is reduced to NADH. Okay, so let's talk about the Krebs cycle. The Krebs cycle occurs like this. The acetyl-CoA that you formed in step 1.5, and how many acetyl-CoA should you have at this point from a single glucose molecule? That's right, you should have 2 acetyl-CoA molecules from 1 glucose molecule, 2 acetyl-CoAs. your acetyl-CoAs enter the Krebs cycle, and when they enter the Krebs cycle, the acetyl-CoA joins forces. It actually gets attached to this molecule called oxaloacetic acid, also known as oxaloacetate. I hope you guys are understanding that molecules that end in ate can also be called ic acid. So right here, oxaloacetic acid is the same thing as oxaloacetate. Citric acid is the same thing as citrate. And do you remember back in the previous step, pyruvic acid is the same thing as pyruvate. So hopefully this helps you in your chemistry classes and your biochemistry classes. Just don't get confused. Pyruvic acid is pyruvate. Okay, acetate is acetic acid. Oxaloacetic acid is oxaloacetate. Citric acid is citrate. So this should help you a lot as you move forward in your studies. But anyway, acetyl-CoA, when it enters the Krebs cycle, it attaches to an organic molecule called oxaloacetate to form what's known as citric acid or citrate. This is why this is also known as not just the Krebs cycle, but it's called the citric acid cycle, because the first molecule you form is citric acid. How? Again, by attaching acetyl-CoA to oxaloacetate, you make citric acid. Now, what's the whole point of this? Why do you make citric acid? Well, because now you want to harvest all these hydrogens. You want to give these hydrogens, mainly the electrons from the hydrogens. Because remember, what is a hydrogen? A hydrogen is a electron and a proton, right? So you want to give these hydrogens to the electron transporters, NAD+ or FAD, right? And the citric acid cycle or Krebs cycle or TCA cycle allows you to do that. Okay, so let's see that in action. The citric acid cycle is changed into the isocitric acid molecule. It's basically an isomer. Then isocitric acid is oxidized, right? So it loses some of its electrons. Those electrons are given to the electron carrier, NAD+. NAD+ becomes reduced to NADH. Okay, and also one of its carbons leaves as CO2. So 2 carbons entered and 1 carbon now leaves as CO2. Next, what's left over after this oxidation event is alpha ketogluteric acid, which is then further oxidized into succinyl-CoA. And again, by the way, you don't need to know every little baby step here. I'm explaining it to you so you understand. And then later at the end, I will tell you what you need to know for my class, for my exam. Okay, so alpha ketogluteric acid becomes oxidized to succinyl-CoA. The electrons are given to NAD+, so NAD+ is reduced to NADH, and again, because of this, one of its carbons leaves as CO2. So 2 carbons entered, 2 carbons leave as CO2. What's left? A bunch of hydrogens. By the way, succinyl-CoA is used it becomes phosphorylated, then dephosphorylated, so this allows you to make an ATP via what process? This ATP is made by substrate level phosphorylation. You form 1 ATP per round of the Krebs cycle. So you make ATP by substrate level phosphorylation. So succinyl-CoA becomes succinic acid, or also known as succinate, right? Now you get the trick, right? Succinic acid, succinate. Succinic acid is then oxidized to fumaric acid. That means that FAD picked up its electrons and protons and became reduced to FADH2. Fumaric acid is then, water is used to modify it to malic acid or malate. Okay, malic acid means malate. Malic acid is then oxidized finally to oxaloacetic acid or oxaloacetate. And in the meantime, NAD+ is reduced to NADH. So you've reformed oxaloacetate at the end of the Krebs cycle. You've made more oxaloacetate. And what does the oxaloacetate do? That's right, it picks up the next incoming acetyl-CoA. And how many acetyl-CoA did you form? But from one sugar molecule, one glucose molecule? That's right, you should have gotten 2 of them. So in the process of the Krebs cycle, you should make 2 acetyl-CoA. Okay, so what do we need to know? You don't need to know every single little step I mentioned, but what do you need to know? Well, you need to know that 2 acetyl-CoA enter the Krebs cycle for every 1 glucose molecule. You should know that acetyl-CoA attaches to or binds to oxaloacetate or oxaloacetic acid to form citric acid or citrate. Okay, you should know that 1, 2, 3 NADH are formed, and 3 NADH are formed, but how many rounds of the Krebs cycle did you have per glucose? That's right, 2. So 2 acetyl-CoA come in, 1, 2, 3 times 2, 6 NADH are formed total from 1 glucose. 2 FADH2 are formed per 1 glucose. Remember, because you have 2 rounds of the Krebs cycle, because you had 2 acetyl-CoA. And 2 ATP are formed as well. So again, 2 acetyl-CoA enter, 6 NADH are formed, 2 FADH2 are formed, 2 ATP are formed by what process? By substrate level phosphorylation. And how many CO2 waste products are formed? That's right, 4. 4 CO2 waste products are formed per glucose. And I'm sorry if I skipped this, but during this half step here, remember oxidation of pyruvate, what was the overview here? 2 pyruvate are used up, right? 2 pyruvate, because remember at the end of glycolysis, you had 2 pyruvate. 2 pyruvate are used up to form 2 CO2 waste products, 2 NADH, and 2 acetyl-CoA. Okay, that was the overview here. And those 2 acetyl-CoA enter, again, the Krebs cycle to make 6 NADH, 2 FADH2, 2 ATP, and a total of 4 CO2 waste products. So if you've been keeping track of the math, glucose is a, let me show you. Glucose is a 6-carbon molecule, 1, 2, 3, 4, 5, 6. 6-carbon molecule, which gets cut in half into 2 pyruvates, okay? Those 2 pyruvates enter this half step called oxidation of pyruvate, where 2 of its carbons leave as CO2. So your 6-carbon sugar lost 2 carbon here as CO2. So how many carbons do you have left over? You had 6 carbons. 2 of them leave as CO2 right here. So you have 4 carbons left, right? And then during the Krebs cycle, the other 4 carbons leave as CO2. Did you see what happened? Glucose is a 6-carbon molecule. When that 6-carbon molecule has 6 carbons, 6 oxygens, and 12 hydrogens, the carbons leave this whole process as CO2, and that's what you breathe out. That's why you breathe out CO2. And the oxygens also leave as CO2. But what you're trying to harvest from your sugar to get ATP is not the carbons or the oxygens from the sugar molecules, but the hydrogens. The hydrogens which you, through redox reactions, handed to your electron carriers, NAD plus or FAD. Does that make sense? It's those hydrogens that harbor the all-important electrons. Those electrons go with the help of the electron carriers, NADH and FADH2. Those electron carriers transport those hydrogens. Let me show you this overview slide here so you know what I'm talking about. Those carriers transport those electrons, right? Those hydrogens, which are essentially an electron and a proton. the NADH that was formed during glycolysis, the NADH that was formed during pyruvate oxidation, the NADH that was formed during the citric acid cycle, and the FADH2 that was formed during the citric acid cycle, they all transport those hydrogens to the all-important last step called oxidative phosphorylation. And what happens is those hydrogens are given to the electron transport chain. Those electrons go through the electron transport chain to power the electron transport chain to pump protons. And those protons allow chemiosmosis to occur to give you your lion's share of the energy, the 28 ATP. Okay, so again, why do I bring that up? Well, I wanted to show you the main point of glycolysis. Like, why is there glycolysis? Why is there pyruvate oxidation? Why do we have citric acid cycle? Well, it's numerous steps with one goal. Get the hydrogens from the glucose. Remember, there's 12 hydrogens on your glucose. Get those hydrogens from the glucose to the carriers. So NAD plus becomes NADH, reduced to NADH. You got more NADH. You get more NADH. You got more FADH2. These H's are the H's that were on the glucose molecule, and these H's get they get transported to the electron transport chain, where they hand those H's, specifically the electrons of those hydrogens, to the electron transport chain. That's what powers the electron transport chain and allows for the electron transport chain to function, allows for oxidative phosphorylation to occur, chemiosmosis to occur, and for 28 ATP to be formed. Okay, so that's kind of how it works. So the only other step left to talk about is the electron transport chain and chemiosmosis, which is known as oxidative phosphorylation. Okay, so let's talk about oxidative phosphorylation. But first, let's take a quick break time with TIG. Hey everyone, welcome back from break time with TIG. So where we left off, I was about to explain oxidative phosphorylation. Remember, please watch the video I provided the link for earlier, where I explained cellular respiration, because I go into great detail about oxidative phosphorylation in that video. You've got the electron transport chain, which consists of complex 1, complex 2, complex 3, complex 4, and followed by the ATP synthase. And I go into great detail of how this whole system works in that video. So please make sure you watch that. Otherwise, everything I say here is just going to be redundant. So I'm going to assume that you've watched that and you know how the system works. And so I can pick up there. All right, so what I want you to know that's in addition to that video is that the electron transport chain and these components, complex 1, 2, 3, and 4, as well as ATP synthase, in eukaryotes, all of these complexes exist in the inner membrane or cristae of the mitochondria. Okay, but this is a microbiology class, right? Do prokaryotes, you know, these bacteria, archaea, do they have an inner membrane of a mitochondria? The answer is no. Prokaryotes do not possess mitochondria. Prokaryotes do not have mitochondria, right? Mitochondria is a membrane-bound organelle, and prokaryotes do not possess membrane-bound organelles. So what that means is, you know, you might be asking yourself, well, where is the electron transport chain of a, let's say, E. coli bacteria, right? Where is the electron transport chain if it doesn't have a mitochondria? Where is the ATP synthase if it doesn't have mitochondria? Well, the answer is here. Take a look. This is a nice slide that juxtaposes what's going on with cellular respiration in a bacterium on the left and what's going on with cellular respiration in a mitochondria, in a eukaryote on the right. Remember, we just covered this in a mitochondria, the inner membrane, which is highly folded, or what they say convoluted. These structures are called cristae. In the inner membrane, you have this electron transport chain followed by ATP synthase. In a prokaryote, all of that's happening in the plasma membrane. So look here at the cell wall. Here you have a plasma membrane, or the cytoplasmic membrane, same thing. And that's where these complexes exist. All right, so what's happening in a prokaryote, this is what I want you to know, is that the protons are being pumped physically out of the cell. So the proton gradient is low inside of the cell because the ETC is pumping the protons out of the cell. You see that? So you have a greater concentration of protons outside of the cell and a much lower concentration of protons inside of the cell. And then that's why by chemiosmosis, those protons will diffuse via chemiosmosis through ATP synthase, which is what causes ATP synthase to spin and what provides the energy for ATP to form, right? Remember energy coupling, and exergonic process will drive an endergonic process, or an exergonic process will provide the energy for an endergonic process. Another way of saying that is a spontaneous process can be used to power a non-spontaneous process. Remember, we talked about that in the previous video I told you to watch. Protons are forcing their way in. That is diffusion of protons, a process known as chemiosmosis, a process which is spontaneous or exergonic. That releases energy. That energy is being used to do a process that requires energy, forming ATP, phosphorylating ADP to ATP. You see that? So ATP is being formed, and that takes energy. Where did that energy come from? That's right, diffusion of protons, which means chemiosmosis. So again, in prokaryotes, this whole process is happening in the cytoplasmic membrane or plasma membrane. In eukaryotes, it's happening in the inner membrane of the mitochondria. So good. Now, one thing I want you to know is the PO ratio, right? I want you to understand how many ATP are being, how many ATP are being produced per NADH, how many ATP are being produced per FADH2. And remember, this is the process here. NADH drops off electrons. It's 2 electrons at complex 1. Those electrons they spontaneously move to complex 4, where they are picked up by oxygen to form water. FADH2, on the other hand, drops off its electrons here, closer to complex 2. Those electrons go through to complex 4, and they're picked up by oxygen to form water. Now, the PO ratio, which I was just talking about, refers to how many ATP are formed for every NADH, or how many ATP are formed for every FADH2. See, at this whole process, the purpose for it is to make this ATP at the end. So how many of these ATP do we get for every NADH that's oxidized? How many FADH2 per FADH2 that's oxidized? Now, here's how you figure that out. They've already figured it out for you. You just have to remember is what I mean. NADH drops off its 2 electrons. Those electrons move spontaneously through the electron transport chain. Spontaneous process releases energy. That energy is being used to pump protons, complex 1, 3, and 4, pump protons into the intermembrane space for eukaryotes, or into the outside of the cell if it's a prokaryote. Those protons then, remember, force their way through ATP synthase, causing it to spin, and that's what provides the energy to make ATP. Now, what you need to know is for every NADH that drops off its electrons, 10 total protons are pumped. 10 total protons are pumped. However, it takes 4. It takes 4 to make an ATP, all right, and provide it to the cell. So what is the PO ratio? For every NADH, 10 protons are pumped, but it takes 4 to make an ATP. So what's 10 over 4? 2.5. So the PO ratio of NADH is 2.5. What does that mean in layman's terms? It means 2.5 ATP are made for every 1 NADH that is oxidized. That means for every 1 NADH that visits complex 1 and drops off its electrons, right? Remember, NADH is the reduced form of the molecule. NADH reduces complex 1, and NADH, in the process of reducing complex 1, NADH becomes oxidized to NAD+. Okay, what about FADH2? FADH2 drops off its 2 electrons. Those electrons go down the electron transport chain. That actually results in fewer protons being pumped. Fewer. Why? Because we're skipping pump 1, aren't we? We're skipping this pump. If you skip a pump, you're not going to pump as many protons. So FADH2 skips pump 1. It gives its 2 electrons to coenzyme Q. The electrons go through the electron transport chain, and that you end up pumping 6 total protons, right? 6 protons. Now, remember, it takes 4 to make ATP, a 1 ATP molecule, to phosphorylate ADP to ATP. It takes 4 of these protons through a process of chemiosmosis coming through the cell. So what's the PO ratio here? It would be 6 over 4, which is 1.5. So in this case, the PO ratio of FADH2 is 1.5. Okay, so please understand the PO ratio. It refers to how many ATP do you obtain per oxidation of NADH or FADH2. Now, last concept for part 1 of this chapter 5 video. Last concept for part 1 of chapter 5 video is this. And that is this concept of fermentation. I want you to understand fermentation. Fermentation is an anaerobic process that occurs when you don't have enough terminal electron acceptor. Okay, I want you to know that fermentation occurs when you run out of terminal electron acceptor. So let me show you what I'm talking about. Remember here at oxidative phosphorylation? Take a look here. What's the terminal electron acceptor here? Remember, oxygen. Oxygen is called the terminal electron acceptor because oxygen picks up the electrons at the end of the electron transport chain. It's where the electrons end up. Remember, the electrons started on glucose. They were transferred to the electron carriers, NADH or FADH2. They were then transferred through a series of redox reactions to electron transport chain. And finally, those electrons make it to oxygen. So oxygen picks up the electrons at the end, at the very end. Okay, that's why it's called the terminal electron acceptor. So this must be what process? This must be aerobic cellular respiration. Remember, if we had anaerobic cellular respiration, what would be the only difference? We already covered this. That's right. There would be a different molecule here to pick up the electrons at the end of this ride, at the end of the electron transport chain. So if it wasn't oxygen that was picking up these electrons, that would be called anaerobic cellular respiration. But because it is oxygen picking up these electrons, that is aerobic cellular respiration. So anyway, all that to say, what is fermentation? Okay, what is fermentation? Fermentation, I need you to understand this. It's very important. Fermentation is not anaerobic cellular respiration. because you already know what anaerobic cellular respiration is, right? I told you anaerobic cellular respiration is when a molecule other than oxygen picks up these electrons at the end of the electron transport chain. Aerobic cellular respiration is when oxygen picks up these electrons at the end of the electron transport chain. So what's fermentation? Fermentation is when you do not have enough, you see here, you do not have enough terminal electron acceptor. So if it's oxygen is your terminal electron acceptor, you don't have enough oxygen. If some sulfur containing molecule is your terminal electron acceptor, you don't have enough of that sulfur containing molecule. Whatever your terminal electron acceptor is, you don't have enough, right? So the example here is you go to the gym and you're working out at the gym and you're running really hard, and next thing you know, everything starts cramping up and you have to sit down, right? What happened? Your body was not getting enough oxygen. Your body was switching from aerobic cellular respiration to fermentation. Okay, in humans, that fermentation process is called lactic acid fermentation. So what happens? Well, if you don't have enough oxygen to pick up the electrons at the end of the electron transport chain, well, then the electron transport chain gets backed up with electrons, right? Like a traffic jam. So you get backed up electrons, and that means that the electron transport chain shuts down, and NADH and FADH2 can't drop off any more electrons because this whole thing is already jam-packed with electrons, and there's no way to get the electrons off the ride. So what does that mean? Let's go back to this overview slide here. Let me show you this slide. So what I was just telling you is the electron transport chain shuts down, which means oxidative phosphorylation shuts down. Not only that, the citric acid cycle, aka the Krebs cycle, shuts down. Not only that, pyruvate oxidation shuts down. Okay, the only thing that does not shut down during fermentation is glycolysis. Okay, I need you to know that during fermentation, you run out of terminal electron acceptor. So, for example, you run out of oxygen. This process shuts down, this process shuts down, this process shuts down. Glycolysis is the only process still ticking. So what that means is you're converting glucose to pyruvate. The pyruvate can't go anywhere. And in the meantime, you're producing 2 ATP and 2 NADH. Does that make sense? So how do we keep this process going? Okay. How do we keep glycolysis going? Let me tell you something. You see this NADH? How did you make NADH? You converted NAD plus to NADH, right? So let's talk about this. Let's talk about how cellular respiration switches to fermentation and how fermentation keeps going and prevents stalling out. Okay. Here, let's start with this one. This is called lactic acid fermentation. This is the process that occurs in humans and animals and many prokaryotes, actually. This is called lactic acid fermentation. So what happened? You hit the gym too hard. You ran out of enough oxygen to keep your cellular respiration keep going. So what happens? You switch to just glycolysis by itself, which means you took a glucose molecule. Your cells are converting the glucose to pyruvate, right? To 2 pyruvate molecules. In the meantime, you form 2 ATP through substrate level phosphorylation. You also form 2 NADH as well, right? Now, that's all good and dandy because you're netting 2 ATP, but here's the problem. I want you to understand the problem here. The problem is every time you make these 2 NADH, they have no place to go during fermentation, right? Normally, what does the NADH do during aerobic cellular respiration? That's right. Normally, the NADH goes to the electron transport chain, drops off those electrons, and once it drops off those electrons, NADH gets oxidized back to NAD+. Does that make sense? And then the NAD+ can come back and pick up more electrons. Did you guys follow that? Normally when you make NADH, the NADH gives the H to the electron transport chain that oxidizes NADH back to NAD plus so that it can come back and pick up more electrons and do it over again. But does the NADH have a place to go during fermentation? Remember, the electron transport chain is shut down. So there's no place for NADH to go. Does that make sense? So in this case, the NADH just keeps accumulating. And when that accumulates, what are you using to make NADH? NAD plus. Look at this. The NAD plus is getting converted to NADH. If there's no way to oxidize NADH back to NAD plus, that means all of your NAD plus becomes NADH. You guys follow me? That's a problem, because the minute you run out of NAD plus, you're going to run out of glycolysis, because glycolysis can't happen without NAD plus picking up those electrons. So if all of your NAD plus become converted to NADH, you're actually going to stall out glycolysis, and that could mean death, right? That's not good. So you need a way of replenishing what molecule to keep glycolysis going? Think about it for a second. Maybe pause the video. Think about it. What do you need to replenish to keep fermentation going, to keep this glycolysis going? That's right, NAD+. You need to replenish your NAD+. Otherwise what? Otherwise all of it gets converted to NADH, gets reduced to NADH. Does that make sense? So that's why you end up with these fermentation products, these various fermentation products. So here's what happens. Glucose. During fermentation, glucose goes through glycolysis, becomes 2 pyruvate molecules. In the meantime, you've reduced 2 NAD plus molecules to NADH. So 2 NAD plus become converted to NADH. And 2 ATP were formed net by substrate level phosphorylation. That's great. This is what you want. But how do we get more NAD plus? Well, NADH can't go to the electron transport chain, so what it does is something clever. It gives this H, those 2 electrons, it gives those 2 electrons directly to pyruvate. Remember, because pyruvate can't go anywhere either. When you reduce pyruvate, it becomes lactate or lactic acid, right? Remember, lactate means lactic acid. So now you've formed lactic acid. And that's why this is called lactic acid fermentation. And that's why when you hit the gym too hard, they say, you know, if you're playing sports and you hit the gym too hard, your coach will say, you need to take a breather because you formed lactic acid, right? You have lactic acid buildup. Okay. Why did your body make lactic acid? Not because your body really loves lactic acid or does anything with lactic acid. It's because pyruvate becomes converted to lactic acid. Pyruvate becomes converted to lactate. Does that make sense? Why? Because you need a way of replenishing NAD+. That's the thing. So your body's not trying to make lactate. It's not beneficial to your body to make lactate. In fact, lactate's kind of toxic to your body. However, it's a necessary evil of producing more NAD+. Okay, so if I were to ask you on the exam, what needs to be replenished for fermentation to continue going, your answer would be what? Not glucose. Yeah, normally your body has plenty of glucose. It's NAD+. NAD+. Okay, great. So NAD+ needs to be replenished for fermentation to keep going. And how do you replenish NAD+? Well, you need to take NADH and reduce pyruvate to lactate. That way your NADH becomes oxidized back to NAD+, and that NAD+ is ready to pick up more electrons. You see? So that's how it works. So quick refresher, when does fermentation happen? That's right, when you are short or you run out of terminal electron acceptor. How many ATP are formed during fermentation? 2 net ATP. What's the only process that occurs during fermentation of the normal cellular respiration process? Glycolysis is the only step that occurs. Only glycolysis, not Krebs cycle, not oxidative phosphorylation. What needs to be replenished during lactic acid fermentation or during any fermentation? That's right, NAD plus needs to be replenished. How do you replenish it? Well, in this case, in the case of lactic acid fermentation, the way you replenish NAD plus is you reduce pyruvate to lactate. Great. Now, there are different forms of fermentation, but they all have the same goal, okay? And they all have the same cause. The cause is the terminal electron acceptor is low. However, not all creatures on Earth form lactic acid. Some of them form alcohol. Some of them form isopropyl alcohol. Some of them form all kinds of things, okay? Acetone. There's many, many different fermentation products. So let's talk about a different fermentation product. You've heard of alcohol fermentation. Some creatures do this instead of lactic acid fermentation. Some bacteria, and namely humans know about yeast, right? Saccharomyces cerevisiae. That's the creature that that's the baker's yeast. That's the yeast that makes beer or alcohol for us. Okay, so some creatures, they do a different kind of fermentation. They have a different fermentation product, right? There are many different creatures out there, and not all of them do the same type of fermentation, and there are many different fermentation products. Our product is lactic acid. Yeast is alcohol, ethanol, drinking alcohol. And then there are other fermentation products out there as well. In fact, let me show you this real quick. Here. This is just showing you the microbes and the fermentation products they make. See, you can make carbon dioxide and propionic acid. That's a fermentation product. Lactic acid is a fermentation product. Ethanol and CO2 is a fermentation product. Acetone, isopropanol is a fermentation product. There are many different fermentation products. But I don't want you to get confused. Why do these creatures switch to fermentation? Because they've run out of terminal electron acceptor. How many ATP are formed during any one of these types of fermentation? 2 net ATP. Why? Because only glycolysis is occurring during all these forms of fermentation. Does that make sense? And what's the limiting factor that needs to be replenished during all of these forms of fermentation? That's right. NAD plus is being replenished. That's why you end up with isopropanol. That's why you end up with acetone. That's why you end up with ethanol. That's why you and I as humans end up with lactic acid. That's why you end up with propionic acid. Does that make sense? So let's talk about alcohol fermentation to round out this part 1 chapter, and then we'll move on to the next video, chapter 5 part 2, where we're going to be discussing photosynthesis. And again, please make sure you watch the video that I've already put together, reviewing what you should have learned in Biology 1406 about photosynthesis, so that chapter is more digestible and easier to understand. Okay, so anyway, let's talk about alcohol fermentation. Again, this would happen in, for example, yeast. Saccharomyces cerevisiae is the scientific name for this yeast. And here's what you do. Glucose, oh, by the way, so why do you think when you're making alcohol, when vintners make wine or beer crafters make their brews, okay, breweries make beers, why do you think that they put the yeast with some kind of sugar and they reduce the oxygen? They keep the oxygen out, right? So if you make wine, you put it in a barrel and you cork it so no oxygen gets inside. Well, because you're trying to limit the terminal electron acceptor so that the yeast switch to fermentation from cellular respiration to fermentation, because you've put a cork in it. You've put a cork in the bottle, right? So those yeast now don't have oxygen or as much oxygen, so they're switching to fermentation. Those yeast are taking glucose, doing glycolysis, netting 2 ATP, and converting NAD plus by reducing NAD plus to NADH. They're also producing 2 pyruvate molecules. However, you and I, as lactic acid fermentators, you and I would directly reduce pyruvate to lactic acid, right? Well, these bacteria and yeast, these yeast that can do alcohol fermentation, the only difference is, first they convert the 2 pyruvate molecules to 2 acetaldehyde molecules. And so basically they cleave this CO2 off. You see, this CO2 leaves as carbon dioxide, and that's why your beer can get bubbly, right? That's why you get bubbles in your beer, right? The CO2 is cleaved off. And then you have a 2 carbon molecule called acetaldehyde. Now you can reduce acetaldehyde. You take the H from NADH because we want to make more NAD+. You take the H from NADH, 2 electrons actually, you give it to acetaldehyde. Acetaldehyde becomes reduced to ethanol. And why did we make ethanol? Why did the yeast make ethanol? Does it really like ethanol? No, it was a necessary thing to make more NAD+. The thing that needs to be replenished is NAD+. Why? Because now NAD+ can pick up 2 more electrons and a proton to become NADH, and the process can occur over and over again. So again, what do I need you to know? In you and me, in humans, we directly reduce pyruvate to lactic acid, but in alcohol fermentation, the pyruvate is converted first to acetaldehyde, releasing CO2, and then that is reduced to ethanol. So I hope you understand why vintners, these people that make wine, need to make sure that no oxygen is getting in there. It's because you want to switch the conditions to anaerobic, you want to switch it to fermentation, and you want to make sure that ethanol is being produced. Okay, so that's how it works. That's cellular respiration. You've learned 3 big concepts, right? Cellular respiration, the difference between aerobic and anaerobic cellular respiration, and fermentation. Again, I want you to know this for sure. This is very, very important. In respiration, you see, respiration, you've got glycolysis, pyruvate oxidation, citric acid cycle, and oxidative phosphorylation. That's what respiration means. Aerobic respiration means oxygen is the terminal electron acceptor. Here at this electron the ETC, electron transport chain. Anaerobic respiration means a molecule other than oxygen is your terminal electron acceptor, whereas fermentation means this is not happening, this is not happening, this is not happening. Fermentation means only glycolysis is happening, because whatever your terminal electron acceptor happens to be, you don't have enough. All right, does that make sense? So please understand all that. Let me know in the comment box below if you have any questions, and I'll catch you guys in part 2.
Dr. D, Dr. D, Dr. D. 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 Dr. D,